dear students. Today we are going to start an interesting discussion on plant growth and development. All plant cells are descendants of zygote, that is a fertilized egg. Descendants, what do you mean by that? That means they are produced from the zygote. So all plant cells are produced from the zygote that are known as fertilized egg. The zygote develops into mature plant through growth and differentiation, forming roots, leaves, branches, flowers, fruits and seeds. Then they eventually die. So in a process, if we see, the zygote develops into a mature plant, but in between the development or full grown conditions, it undergoes growth and differentiation. So during this process, that means during the growth and differentiation process, they form the roots, leaves, branches, flowers, as I told you just earlier. So now you see the diagram below. First cell, that is a zygote cell, which is formed of a mature egg, and after fertilization with the sperm, then it becomes the two cells conditions, then 8-celled, then globular embryo, heart-shaped embryo, and gradually it forms the mature plants. So during all these processes, they have to follow the development and differentiation process properly. So now we are going to discuss about the plant growth. Let's see what are the points here. So growth is an irreversible permanent increase in size of an organ or its parts or an individual cell. That means after growth, it cannot be reversed. Once a zygote becomes the mature, it can produce another zygote, but it can't revert back to the zygote. So it involves metabolic processes that consume energy. During this growth processes, what happens? Metabolic processes, they consume much more amount of energy. Now you see, plant growth generally is indeterminate. Plant growth continues throughout the life due to the presence of meristems. Because they possess meristems, a special kind of tissue, they are capable of divide and redivide themselves. So meristematic cells have the capacity to divide and self-perpetuate. And the growth, where new cells are always added to the plant body by the activity of the meristem, is called open form of growth. So generally plants grow throughout their life, which is not happening in the animal cells. You see, primary growth and secondary growth, two type of growth usually we are looking in the higher grade plants. It occurs due to the primary growth occurs due to the root apical meristem and shoot apical meristem. That means the cells or tissues found in the apex, either in the root apex or in the shoot apex. It causes the elongation of plants along their axis, and due to the divisions of these apical meristem, the plant grow along the axis and increase its length. Now the secondary growth in gymnosperms and dicots especially it is seen. It occurs due to lateral meristems. Vascular cambium and cork cambiums are the tissues who are taking part in the secondary growth. That is also an important section. You can deal with that in another section, another chapter. I will definitely supply this to you. So This causes the increase in girth of the organ. So it increases the girth, that means the width of the plant, you can say, and you see the primary growth is increasing the length of the plant, whereas the secondary growth is increasing the girth of the plant. Now let's see, growth is measurable, that can be measured. Is at cellular level, growth occurs due to increase in the amount of protoplasm. If we are looking the growth in a cell, then we will find that the protoplasm is increasing. So increase in protoplasm is difficult to measure directly. It is not so easy. So growth is measured by parameters like increase in fresh weight, dry weight, length, area, volume, and cell numbers. So these are the parameters through which we can measure the growth. So cell numbers, a maize root apical meristem can produce more than 70,500 new cells per hour. So if we see in experimental conditions, a maize root apical meristem can produce above 70,500 cells, new cells per hour. If we are looking at the cell size, then cells in a watermelon may increase in size by up to 3,50,000 times, and a single cell can increase such a large size. Similarly, if the length is seen, then growth of a pollen tube is markable. Now the surface area, growth in a dorsiventral leaf can also be marked. So these are the measurable situations of the different plant parts. Growth can be measured in such a way. Next, you see, three phases of growth are there: meristematic phase, elongation phase, maturation phase. So let's see first what is a meristematic phase. It occurs in the meristems at the root apex and the shoot apex. Cells in the regions have rich protoplasm and large nuclei. Cell walls are primary, thin, and cellulosic with abundant plasmodesmata. These cells are very active because they possess more amount of protoplasm and have an active nuclei, and very shortly they can divide and redivide to produce their new cells. So these are found. Meristematic phase can be seen in the apical region where growth is well marked. Now you see the elongation phase. It occurs in cells proximal, just next away from the tip. That means just behind the apical region. You see where the meristematic zone is there. So just behind the meristematic zone, the cells increase in their length and the cells have increased vacuolation, size, and new cell wall deposition. So gradually the cell increase in size during this phase. And the maturation phase, it occurs in the cells further away from the apex. Again it is from the further away from the apex, more proximal to the phase of elongation, and the cells attain a maximal size in terms of wall thickening and protoplasmic modifications. Any kind of modifications, length growth, that can be limited there. So after maturation phase, there may be the end of the growth, we can say, for the plant cells. Gradually they can degrade. Next we see growth rates. So it is the increased growth per unit time, arithmetic growth. In this following mitotic cell division, only one daughter cell continues to divide while the other differentiate and matures. So after the mitotic division, only one daughter cell continues to divide. That means out of the two product, one is dividing regularly and one is entering to the differentiation and becomes mature gradually. So on plotting the length of the organ against time, a linear curve is obtained. When we are drawing a graphical structure by taking time and length of the plant or growth, we can say, we will get a linear curve, and this explains the arithmetic growth of the plant. Similarly, another policy can also be seen, but now you see. So mathematically it is expressed as arithmetic growth can be expressed as LT, that is equal to L0 plus RT. So here LT represents the length at time t, where L0 is length at time 0, that means at the time of beginning of our measurement. R is the growth rate, that means elongation per unit time, definite time. In definite time, how much it is growing, that is representing R, and the T represents the time. So this is all about the arithmetic growth. So we can see also growth rates in geometrical fashion. This is also called geometrical growth. In most systems, the initial growth is slow, that is lag phase, LAG, lag phase. Then it increases rapidly, and it is called log or exponential phase. Here, both the daughter cells continue and retain the ability of mitotic cell division. If nutrient supply is limited, the growth slow down and leading to a stationary phase. So in this pattern, that means in the geometrical growth, we are looking that initially the growth is a little bit slow, but gradually it increases very rapidly, and if nutrient supply is limited or any other growth factors is going to be limited, then the growth leads to a stationary phase, and it forms a different graphical structure that is looking like a S. So geometrical growth, when we are plotting graph, taking the parameter of growth against the time, we get a typical sigmoid or S curve. So S curve is representing the geometrical growth. A sigmoid curve is a characteristic of living organism. In all living organisms that can be marked growing in a natural environment, it is typical for all cells, tissues and organs of a plant. That means you can see it is more practical than the arithmetic growth. The exponential growth can be expressed as, this exponential growth can be expressed as W1, that is equal to W0 ERT. What is W1? That is equal to final size, that represents weight, height, number, etc. Similarly, W0 is the initial size at the beginning of the period when we are starting our study, and R is equal to relative growth rate. So here T is also time of growth, and E is representing the base of natural logarithm. So you can be asked to write a 2 marks questions both in geometrical growth and arithmetic growth. I hope from this section you can also write. Now we will discuss about the growth rates. Relative growth rate, or R, is the measure of the ability of plant to produce new plant materials, referred to as the efficiency index. Hence, the final size of W1 depends on the initial size, that is W0. So quantitative comparisons between the growth can also be made in 2 ways. Number 1, absolute growth, measurement and comparison of 2 growth per unit time. and number 2, that is relative growth rate, measurement of growth of the given system per unit time expressed on a common basis per unit initial parameter. So in this diagram you see there are two leaves, a comparative diagram, diagrammatic comparison of absolute and relative growth rates. Both leaves A and B have increased their area by 5 cm square in a given time to produce A1 and B1 leaves. Look at this. In A leaf, initial size was 5 cm square and the final size is 10 cm square. But in the B leaf, it was earlier 50 cm square and it grows up to 55 cm square. So this is the comparison of absolute and relative growth rate. Now you see what are the conditions, essential elements for growth. They are water, oxygen, nutrients, temperature, light, gravity, etc. You see, water for cell enlargement or turgidity of cells, that means the hardness of cell that is brought out by the deposition of water, helps in the extension growth. So water provides medium for enzymatic activities needed for growth. Oxygen, it helps to release metabolic energy for growth. Macro and micro elements are needed for the synthesis of protoplasm and act as source of energy. Similarly, temperature, plant have an optimum temperature at which growth is maximum. Deviation from the range could be detrimental to its survival. That means if the temperature is deviating towards low or high, it may cause the death of the plants. So light and gravity affect certain phases or stages of growth. So these are all conditions under which growth can be maintained. So this is all about the plant growths that you can study in your syllabus. You can have a preparation for 2 marks and 1 marks category questions from this. Hope you will prepare well.